So recombinant protein expression is a method by which we can utilize the protein production machinery in an organism. Usually it's going to be E. coli or yeast, although there might be some other options out there. And the goal is to produce large quantities of a desired protein. And we're going to use a DNA plasmid to accomplish this. And so we can actually change the sequence of our target protein however we want. We can introduce mutations to make a mutant protein. We could build in useful features like affinity tags, things that help us purify the protein, maybe things that help us do a Western blot by adding maybe an epitope or something like that. So lots of options with recombinant protein expression because we essentially can control the sequence of the DNA that we're putting into the organism. Now, control is an important concept when it comes to growing proteins, because there could be some issues. If we give a organism like E. coli the gene to produce a protein, we want to maximize the amount of that protein that we produce. And the issue there could be, well, maybe that protein is toxic to the cells, and if the organism is producing that protein, maybe it kills the cells. And the other thing is, if the organisms are putting a lot of their energy into producing that particular protein, they might not be producing a lot of cells, or they might not be reproducing. And so we want a large cell mass. So we want the cells to grow, and then we want to, at some time later on, initiate, once those cells have reached an appropriate density, we want to then initiate the production of the protein. And so this is all what we would call inducible. We need an inducible system, a switch basically. So we can let the cells grow and divide so they can put their energy towards that. And then when the time is right, we're going to turn on protein synthesis. And so we use elements from the lac operon, which you may have learned about before. The lac operon is a fantastic biochemical switch. And so just as a quick review about what the lac operon is, if you remember, there are these genes that are involved in the biological utilization of lactose, which is something that yeast and other organisms can use as food, but they don't encounter it all the time. And so rather than always having the machinery to utilize lactose, it makes sense to turn that off when it's not needed. Right before that, we have an operator, which is a sequence of DNA that is capable of binding something we call a repressor. The repressor is called lac-I. It's a protein that sits on that operator sequence, and it prevents the transcription of these genes. The promoter sequence is right before that, and that's what's going to recruit the RNA polymerase. But RNA polymerase will encounter this repressor sitting on that operator sequence, and it will not produce the messenger RNA for these genes, and the protein products do not get made. But if we introduce to the system an inducer, and let's say then E. coli or yeast or whatever encounter some lactose out in the environment, that lactose, or actually a product of its utilization, will bind to the lac-I, that repressor protein, and change its conformation such that it can no longer bind to that operator sequence. And so since the operator sequence is open, then RNA polymerase can be recruited by the promoter, and then the genes will be transcribed and then ultimately translated into the products that are needed. Then once the lactose is used up, it will unbind from the repressor. The repressor will rebind and prevent the production again of those genes. So this is a very intricate biological system that evolved. And what we do in the laboratory is utilize some of these control elements to turn on our protein production when we want to. So to show how this works, we're going to use what we call the T7 expression system. And this is where we take elements from the lac operon. We're not taking the entire lac operon. We are not using, the transacetylase and the beta-galactosidase and those structural products. What we're doing is the control elements. We're going to use them to turn on the gene of interest here. So what happens is the T7 system is an engineered system, and it uses a viral promoter. So T7 is a virus, and the promoter then, is a stretch of DNA sequence that's going to recruit T7 RNA polymerase. Now, T7 being a virus, well, E. coli doesn't natively produce T7 RNA polymerase, and the regular native RNA polymerase that E. coli has will not recognize the T7 promoter. And so putting this ahead of our target gene, as we'll see here in a minute, lets us control things quite effectively. Then we're also going to utilize these LAC elements, the LAC operon elements down here, and we're going to use this compound IPTG to be as a stand-in for lactose. So notice how this has a galactose portion here, and instead of being attached to the glucose portion, it's instead attached to this thioisopropyl group, and so this is isopropyl galactopyranoside, or IPTG. So let's see how we can put all these elements together and control the expression of any target gene, say for example AMP-C, which is a beta-lactamase. So this occurs in an E. coli cell, and in this case we're talking about the BL21DE3 E. coli cell. This is a laboratory strain that is particularly good at protein expression. And this has been engineered in a couple of different ways. The major one has to do with its genomic DNA, shown right here. There are things that have been sort of added to its genome. So something that's already present is the native RNA polymerase, that E. coli has. That is, of course, going to be produced naturally anyhow. And this LAC repressor has been added into the genome of the E. coli, and it's under the control of an E. coli promoter, ECP. So under normal conditions, the LAC repressor protein is being produced. Now over here, we've also added into the genome a T7 RNA polymerase. This is the RNA polymerase that came essentially from that virus, and it is sitting ahead of an E. coli promoter. So the native E. coli RNA polymerase would recognize this promoter and could potentially produce the T7 RNA polymerase. However, the LAC repressor will sit on the operator sequence and will prevent the expression of this T7 RNA polymerase. So under sort of normal conditions, there's no lactose or IPTG around, the T7 RNA polymerase does not get made. Only the native E. coli RNA polymerase is present. So remember, we've done a transformation reaction in which we have placed the vector, PET28 in our case, containing our gene, which in this example is AMP-C. So AMP-C is present in this vector. We utilize kanamycin to ensure that only the organisms that contain this plasmid survive. This plasmid has in it also a LAC repressor protein, LAC repressor gene that can be turned into a protein under the control of an E. coli promoter. And the target gene that we're interested in, the protein we wish to express in this example, AMP-C, sits ahead of both a LAC operator sequence, so the LAC repressor will normally sit on that operator and prevent any kind of transcription, but it also sits ahead of the T7 promoter. Now the T7 promoter is, of course, a DNA sequence that the native RNA polymerase in E. coli will not recognize. Only the T7 RNA polymerase will recognize that. So in sort of the default state, when we just start growing these E. coli cells, these transformed cells, this plasmid's present. It's producing the kanamycin resistance product. It's producing the LAC repressor. The organism is not producing any T7 RNA polymerase, and because of that, there's no target protein being synthesized to begin with. However, once the cells reach the appropriate density, what we'll do is add that IPTG molecule to it. And we could add lactose, but the organism could use lactose as a food source, and so the lactose concentrations would decrease. But if we add IPTG, it's going to mimic lactose, but the organism can't actually use it as a food source, and so its concentration stays constant. So if that IPTG is added, then it's going to bind the LAC repressor, which would be sitting on this operator sequence right here. And then the E. coli promoter would recruit the native RNA polymerase, and the T7 RNA polymerase would then be transcribed and translated. So we would now have T7 RNA polymerase present in the cell. That T7 RNA polymerase then would recognize the T7 promoter on our plasmid, And again, because there's IPTG present, the lac repressor would fall off of the lac operator sequence, and this would be open and ready to transcribe our AMP-C gene and ultimately produce the AMP-C protein. So that's the process of how the T7 expression system works to give us an inducible expression of our target protein. So I want to return to that multi-cloning region of PET28 and just show you a few things that are present in there. That has many restriction sites that are available. So you can see all BAM-H1 sites, Hindi3 sites. So there's lots of restriction sites, and what we'll end up doing is putting our gene of interest somewhere in this multi-cloning region. This has many useful resources, such as a hexahistidine tag that we'll see is going to be useful in purifying our protein. It has a T7 tag as well, which can be used as an epitope for Western blotting. And it also has another his tag here on the 3' end of the gene. And the reason for this is we could potentially, depending on how we clone in our gene of interest, we could put a his tag on the 5' end of the gene, which is of course the N terminus of the protein, or we could put it toward the 3' end of the gene, which is of course the C terminus of the protein. So we get a little bit of ability to customize how we build our protein of interest later on. As I mentioned, there is the lac operator sequence right here, which is before wherever our gene of interest is up ahead of it, and the T7 promoter right here. So here's the sequence of that T7 promoter. The ribosome binding site, as we've talked about before, is in this position, and it is several base pairs away from where this start codon is, and that's where the beginning of the protein with the his tag is. So again, just remember what would happen is, in the default state, the lac repressor protein would be binding to the DNA in this position at the lac operator sequence. And before IPTG is added, the native E. coli RNA polymerase would not recognize this T7 promoter, but upon the addition of IPTG, the lac repressor will unbind from the operator, and that will trigger the expression of the T7 RNA polymerase, which will then bind here and begin the transcription and ultimate translation then of our gene and protein of interest. So I just want to go through a couple of steps for the sort of general procedure, the way we would express our particular protein in E. coli. The first thing is to produce plasmid containing the gene of interest, and we've already done that, and you've isolated that plasmid. We then need to transform the cells with that plasmid, and so that's where we're going to transform those BL21 DE3 cells. We're going to grow the cells to mid-log phase in liquid culture with the antibiotic. And just to think about what we're looking at here, let's look at time on the horizontal axis, and then maybe the number of cells, or we could look at this thing we call OD600, basically how optically dense is a solution at 600 nanometers. And so when we first start growing, the cells are getting used to the media. Maybe they were grown in a different medium before, maybe they were on a plate before, maybe they were in a saturated culture before. And so initially, they're not really growing much at all. So they kind of start off kind of lazy like that. But then as they adapt to the new environment, they start multiplying, they will start to take off like this. And then eventually they'll start consuming a lot of the nutrients around and producing a lot of waste, and the culture becomes saturated, and so it kind of tapers off. And then if we wanted to keep going, we're saying number of cells, but eventually we could, the cells could die off and lyse, and then this would go back down. What we want to do is add the IPTG right around this mid-log phase. So that kind of means somewhere around here, when the cells are very active in terms of growing and their metabolisms are active, but they haven't reached the saturation point. If we add IPTG once the cells are saturated, there's not going to be enough nutrients present to produce our protein of interest. So we always want to induce somewhere around here. And then we let the cells continue to produce the protein for some amount of time. Could be a couple hours at 37 degrees. Sometimes we will cool the cells down and let them express that protein overnight. Kind of depends on the particular protein of interest. And then ultimately we harvest them by centrifugation and we get a cell pellet that we can utilize in a protein purification process.